So I'm Hannes Lohi. I'm a professor in molecular genetics. I belong to the Faculty of Veterinary Medicine and also to the Faculty of Medicine and also Folkhälsan Research Center. And my research is really focused using dogs as a model for the human disease. Dogs and cats, a little bit work on horses, cows, but mainly really dogs as a spontaneous model for the human disease. And it's a lot of genetics, but genetic research requires, very good phenotyping and all kind of background information so that you can actually, at the end of the day, identify the genes. So partially, the metabolomics has come to help as well because, it's a tool to phenotype your cases and controls and stratify and classify them in the right group so that you can actually find genes and things. So today, I'm going to tell something about the metabolomics efforts that we have had. And I will start with the disclaimer. So the technology I'm talking about today is NMR-Metabolomics platform. It was first developed by the PetBiomics company in Finland and nowadays PetMeta Labs. So if anybody's interested, so you should contact the PetMeta Labs, but PetBiomics, PetMeta Labs are working closely to each other. PetMeta Labs is partially owned by PetBiomics and then also the Nightingale Health. And I'm the chairman of the board of both PetBiomics and PetMeta Labs, so that's my disclaimer. But really the mission we have jointly is really to enable the preventative, powerful, personalized pet care and diet for dogs. So hopefully this works at one day, which I believe. A little bit about my background. So I was working, and I'm still working a lot about the genetics, and I was involved in a company called Cooper Laboratories, and in that company, that exited 2017. But in that company, we also changed the paradigm. So when I started to do the genetic studies, we were actually looking at single genes, and all the tests in the market were single gene tests. But when you do the genetic research, eventually you start having, gene findings all over the chromosomes in dogs. So it's not that convenient anymore to look at only one region of the genome. So our vision was that why don't we actually develop a technology that you have a single sample and you can measure throughout the genome and gather all that information about the disease risks, health, also about the genomic structure, diversity, and combine that into a nice reportable way to the owners and breeders so that they can utilize this genomic information for better breeding decisions and for vets for veterinary diagnostic purposes, etc. So we developed this kind of 23andMe type of test for the dogs, MyDog DNA test. And this was actually then incorporated later to the Mars Pet Care Wisdom Health collection, and it's actually most sold genetic test today in the world. I think they have tested more than 5 million dogs right now. But that's a little bit about the background. So vision here was go from a single things to more holistic view. And I think the same thing is going to happen here in metabolomics as well. So if you look at the current clinical chemistry, people are measuring few markers, cholesterol, glucose, some other markers, but typically quite a few. And it gives you a limited view what's happening in your body. If you take the serum sample and you measure a few markers only. I mean, they are still very useful and good for diagnostic purposes, but what happens if you could actually take a much broader view, like we took from the genome of the dogs, genome-wide view. So it really changes the paradigm and the view how you actually start seeing things, and it changes the diagnostics and also things. But I think in a longer term, it will improve the health and welfare, a lot of things. So these are my topics here. So I will start about the metabolomics platform that we have, and then I will go to the science behind it. So basically the science means that is this metabolomics platform working or not, and can we actually trust the results or not, and maybe some exciting first result to show the potential of this platform. This technology is based on the human NMR metabolomics platform that has been developed by Nightingale Health, and I think they started something like 2015 or something, and it's the only, I guess, NMR metabolomics platform that is in clinical use. basically worldwide. In human platform, they have optimized 250 biomarkers. And when we were developing this platform for the dogs, you will see here in this slide all the biomarkers for the platform. We have 123 markers for dog. So 250 for the human, but 123 for the dogs. But I think what's important here is that you'll see that these markers, they cover very different type of molecular groups. We have amino acids here. We don't have all the amino acids here, but we have many amino acids and their ratios here. We have lipoprotein particles. Particle sizes can be measured. We have 8 different lipoprotein subclasses here: HDL, LDL, VL, LDL, etc. We can look at the fluid balance with creatinine, albumin. We have glycolysis markers here. And I think what is unique and actually, I would say even revolutionary here is this chronic inflammatory marker, glycoprotein acetates, which has been extremely useful in human platforms and first time now for the veterinary medicine. Then we have, of course, cholesterols in different forms, triglycerides, and fatty acids, for example, omega-3, omega-6 here, and others. Well, if you take a blood samples, there's thousands of compounds, so this is, of course, not detecting everything. So the limitation in NMR is that, it depends what kind of big machine you are using and how many hertzes the NMR machine is having. So typically these measurements are done with 500 megahertzes. You can buy bigger ones with 600 megahertzes. You could then optimize perhaps a bit more markers, but you can never really go up to thousands of markers like in maybe some other platforms like mass spectrometry. But these are the markers. But if you think about the clinical chemistry, now suddenly instead of 4 or 50 markers, suddenly you have actually 123 biomarkers. So now if you have a nice cohort of dogs with different type of diseases or conditions or normal, healthy, or by dogs with different diets, it would be interesting to see what happens to these biomarkers. Are they going up or down or etc.? So that's basically what we have been doing. So here's a little comparison about this technology to the other technologies that are available. So this is the technology here, and I think these are the advantages, I think, what we have. So single blood sample, 123 biomarkers. This is quite quick, one measurement takes about minutes. This is unusually broad data here, not thousands, but 123. In volumes, this can be done with low cost. This is highly accurate, and I think I have to mention here that the consistency and accuracy here means that our results are quantitative. And this is actually a very unique feature in any platforms, that you get quantitative results. So for example, the very much used mass spectrometry, you don't get quantitative results, which is a big problem because you want to really know what the concentration in terms of millimolar, so percentages, is in that animal at the time of the measurement. And this is highly scalable. So I think it's quite useful. Of course, every clinic has different chemistry analyzers there, and they are useful for measure single markers, etc. But to really start working in terms of metabolomics, I think this NMR is quite handy technology there. Now, if you have 123 biomarkers and you have flexible, robust, quantitative technology, what can you imagine? So sort of this picture shows you, maybe something that you could dream. So, and it might enable, so perhaps transition from acute response to the preventative healthcare. I would say that maybe 95% of the vet clinic visits nowadays are acute cases. The dogs really have some problems, or then they come for vaccination or something. But you take the dog to the clinic often too late. So can we change that paradigm towards more preventative healthcare? So maybe this technology allows you to do something about that. I think we have enough coverage to really look at personalized things, personalized medicine, personalized diet and lifestyle changes, and the accurate early diagnostics. so of course required, and this monitoring. So I would imagine where this technology would be in the best use perhaps would be like a regular monitoring, like once a year you get the animal into the clinic and you look at what happens in terms of these biomarkers. And then you look at if something is going out of range. So the red ones here are examples of maybe some biomarkers that are going out, and then you have time to reactive. So you could actually develop like maybe a traffic light model or something that whether your dog has any risks, some diseases. And then you can react and either change the nutrition or lifestyle or medicine for that animal. And if you had animals that have been measured, over the years, you start building up an interesting profile, what happens to the metabolomics in that animal. So the samples that will be used in the technology are serum or plasma samples, and you only need 100 microliters of that sample. And those are typically fasting samples. Yes, I will tell. So that I think this technology really allows, quite interesting visions, if proven that it clinically really works. So if you want to compare the genomics and metabolomics, here's some facts. So the genomics, this is stable, like we have this genetic test, it's static. So you can do one test and, it will be the same results for the rest of the life. But metabolomics is quite different because, it changes. It changes quite quickly. It's dynamic, you eat something, your metabolomics changes. You get the medicine, it changes your metabolomics. You get the disease, it changes. So even during the day, you have quite different metabolomics in your body. And why? Because this metabolomics, reflects, the combination of your genetic and environmental influences in different levels. But this metabolomics layer here, this is actually closest layer to the phenotype here. So if you really want to see what's happening right now in this animal, metabolomics is a handy way to see what's happening in this physiology of this animal here. So I think it's quite unique approach. Perhaps if you want to be really, really good, optimal case for this dog would be that you have this genomics on your record. So you would know what kind of genetics composition this animal had. And then you have regular metabolomics on top of that. So then you could actually really build like a powerful program and follow up the health of this animal. So this technology was based on this human technology, and we have a publication on that. You can access it here. Right now we have analyzed maybe over 13,000 samples from more than 200 different breeds. We have close to 15 publications actually nowadays and 7 years of this platform development. So not very quick program. There's one PhD about this platform development, Claudia Otka, nowadays Claudia Elo. Fantastic PhD here, and I think really showing the potential of this technology. Just the collection of the publications that we have had here. So I think if you really want to have a disruptive technologies for healthcare, you really have to. It has to be backed by the science, and there has to be real findings and things. So we have tried this technology in different cohorts of samples and seeing what happens. And I think I had to say that any cohorts we give to this technology, it gives very, very exciting results. And it gives you a lot of results because you have 123 biomarkers and many of them are changing in different conditions. But I will highlight some examples here in the next few slides. So this was the validation paper. I'm not going to go to the details. If you dig into this paper, you'll find that it's really very well developed technology. So we did this by the book really. We were looking at we collected over 8,000 samples and we were really looking at factors that are affecting, for example, the logistics of the samples. So we were, for example, measuring the same sample every day over 30 days, and we were following whether the results are stable and consistent, and yes, they are. And so you can basically handle the sample as any serum samples you would handle in a clinic. So if you take a blood samples, please spin it down in a half an hour or something like that, because if you don't spin it down, then there's a massive glycolysis going on because of the red blood cells. But this is, the same with any clinical chemistry samples that you take. But it was important for us to look at, because we have different groups of molecules here. Amino acids are actually quite sensitive, so we were actually keeping some of the samples, for example, in room temperature or fridge or -20 or -80, and we were following over time how stable those markers are. And so there's a lot of discussion about this type of, technical logistical things in this paper, so please look at that. But 8,000 samples were collected, around 200 different breeds of dogs in Finland, so a lot of Finnish dog owners participated in the developmental phase. So this is just a reminder about the markers. Then we have looked at different aspects of these things. We were actually looking at what affects, the metabolite profile. So out of those 8,000 dogs, we selected about 2,000 dogs. And these are all healthy dogs. And we were looking at, what are the major factors that are affecting the profiles. And we found, of course, the age. I will show, next slide will show breed has a difference here. For example, the greyhounds, they have much higher creatinine levels. This is not a big surprise because, they are muscular. It's a very muscular breed. Females and males have some differences, particularly in some type of markers like lipids, and of course the diet. If you eat a sausage, you will find a lot of cholesterol in your blood very soon. But these type of things are very important to understand. And here I mentioned that, for example, this GLYCA, this chronic inflammatory marker here, if you look at how it changes by the age, you'll see that, at the age of 14 years, and this is based on all 2,000 dogs in different breeds, at around 14 years of age, it goes out of the, the general range of this biomarker. So when you're doing metabolomics, you need to be careful also about, the age of the dogs. You perhaps have to look at what breeds you are working. All these values are within the range here, but you can see that there's still differences in these different breeds, males and females here, and then different diets here. So particularly, the dry food diet changes the metabolomics a bit. But we haven't really looked very deeply into the diets so far. I will then show some examples about the different conditions that we were working. So here we have a hyper and hypoadrenocorticism. And you can see here, basically the Cushing syndrome samples here, and then the addition disease here, and control samples here. But it looks like, our technology could really, and can actually quite nicely classify these two different conditions and diseases. So if you then look at what are the markers that are driving, this clustering here, you will see that, the addition disease markers here are quite different what you see if you look at the same set of markers in the Cushing syndrome here. So the Cushing is actually here in the blue one, and then addition samples here in the red one. So they're quite opposite to each other. So they are physiological and metabolically quite different diseases. But the interesting thing is that if you have a patient coming to the clinic and you pull out the serum samples, could you actually use this technology to start diagnosing these and putting these into different categories in a nice way? Of course, these results also tell perhaps something about the pathophysiology of the disease, because you see that there's a lot of important markers that are out of range here. We were looking at, and these studies that I'm presenting here are actually based on very small cohorts of samples, which means that we typically had, 25 to 50 cases and then about the same number of controls. So we did not have massive, but even with small number of samples, you see actually quite strikingly interesting results. So we had two different categories of hepatopathies. So ALAT positive, so high liver enzyme activities are shown here in the green, and then the portosystemic shunt here in the blue, and then the controls. And if you look at the markers that are driving again this, there's some amino acids here, palmitic acid, etc. Again, you get quite nice clustering. There's some overlap here, but so really the question here, if we could replicate this in a larger sample cohort, could you actually, if you suspect that this patient has some type of liver disease, maybe it's perhaps you can start clustering them better. And for example, the portosystemic shunt is not always so easy to diagnose. So I think there's a promise there as well, but this is again still small cohort and single samples, but I'm quite excited. We were looking at the epilepsy samples, and so we had dogs that had epilepsy and dogs that were treated by phenobarbital because of this epilepsy, and again very interesting results, broad-wide metabolomic changes, which we could distinct that are not caused by the drug itself, but maybe the disease. So perhaps maybe optimizing the treatment plans could be also beneficial for the dogs. Here an example of one study that we published together with the Walk. They had again epilepsy cohorts, and they were treating them with Purina specific epileptic type food, and so again quite dramatic changes in the metabolomics with treated and non-treated animals. So I think there's clear promise here that perhaps this technology allows you to modify these and make them even better. Here's an example about perhaps liver diseases. We did not actually have really good phenotypes. We have another ongoing project where we have very good kidney phenotypes. So this is a chronic kidney disease, but typically what you see in a chronic kidney disease is a high serum creatinine concentration. So these were basically leftover samples that we were measuring here, but very likely most of the dogs had chronic kidney disease. And as you can see, one of the important markers that's coming really up is the citrate. And we know that in a chronic kidney disease, the blood pH balance is out of range. So perhaps you could actually have a dietary intervention to balance the pH range in these affected patients and alleviate maybe some of the symptoms in these animals. So again, quite interesting results. Typically what you see in these results, it's not the single marker that's popping up, but a series of markers. And perhaps looking at the combination of them is really powerful for the diagnostic purposes, as I will show later. We were also looking at, what happens to the metabolomics during the pregnancy. So we had really carefully characterized small cohorts, but very carefully characterized cohort in early pregnancy, mid-pregnancy, late pregnancy, and then afterwards. And this is just showing, how the ranges are varying over the timeline. But I think again here, this might give you some idea what kind of diets might, balance, give you very healthy and good nutrition for pregnant dogs. And overall, very interesting physiological insights, what happens actually during the pregnancy. We also did another study with 800 dogs. So when we collected these 8,000 dogs to optimize the technology for the dogs, we had about 800 dogs where the owners reported that dogs is either pregnant or pseudo-pregnant or something like that. So we had cohorts of these and we were looking at them. And this technology, a combination of 2 or 3 biomarkers, can actually distinct the pseudo-pregnancy from pregnancy. So you don't necessarily need ultra. You could just take serum samples and run this technology and then see if something is happening. My last slide here. So I mentioned that we have some clinical collaborations going on because the previous results I was showing in the previous slides were based on really more like leftover samples with not very good clinical data. So we wanted to set up a few interesting projects where we actually use other technologies and other routine laboratory values to really do good diagnostics and then compare all that with our technology. So one of the areas that we wanted to study is inflammatory conditions. So we had 7 of them. So this is a submitted study. So we had 7 different conditions, and we had 207 dogs, 175 healthy controls in that group. And so we had atopic dermatitis, enteropathy, kennel calf osteoarthritis, tumors, skin tumors differently, other tumors here, parodontite, pyometra, and urinary infections. So these were collections. There could be potentially more as well, but we focused on these this time. We ran through the samples, so 100 microliters of serums were collected in these animals, and then a lot of different statistical approaches to understand what happens. And this is typically the results that you get when you look at all these markers. And the different colors means that you have different concentrations of these parameters. So for example, here in glucan, you will see, so the 7 different conditions are listed here. So you will see that this chronic inflammatory marker glucan here is really highly upregulated in pyometra, where there's of course very acute massive inflammation. So it picks up, it detects the inflammation very well, actually all over these diseases, not so well maybe in a dermatitis, but there the albumin was, lowered in this case. So, and then you can go through all these markers. So you can actually see that in different diseases, for example, the pyometra has its specific, metabolomic fingerprint. Then in tumors, you have a different fingerprint, etc. So, but lots of changes, you'll see some amino acid changes in different conditions quite dramatically here. So it seems that, this test works both in acute and chronic diseases quite well. And, and we were able to actually develop a model. So the idea was to really see how well this performs in inflammatory conditions. And we found out that the glucan is a fantastic marker, but actually if you take a 6-marker model, the AUC values are actually quite excellent here, so 0.82. So if you have a AUC value 0.5, there's no value in this test. It's just random. But if you have 1, then it's full prediction. But typically in clinical chemistry, values are somewhere 0.65. But so these are really great values, and depending on the condition. So if you look at, for example, the pyometra, the predictive power is close to 100%, if you take these 6 biomarkers. So you can start taking these, fingerprints and, perhaps start diagnosing much better. But at least what happens here is that our metabolomics platform can really identify very powerfully, the inflammatory component in different diseases. And this time we tested 7 diseases. This slide, we compared also, the CRP, which is non-acute marker, cannot be really used for chronic inflammation. So here you see the chronic diseases, the CRP is not really, these are the AUC values. So you can see that, CRP is not working for chronic diseases. It works nicely for acute diseases. But then if you look at the metabolites, they work for both chronic, of course, very well, and then also for the acute. But the prediction power is even better than the CRP. So I think this glucan in combination with some other markers in this platform makes it fantastic for any inflammatory diseases. And we know that there's, 40, 50 diseases at least where we have a very strong inflammatory component. What is glucan? So glucan is a component of, acute phase proteins. So important one is alpha-1 acylglucoprotein, there's haptoglobin, and some other ones. So it's a combination of all these markers. And this one, links some of the conditions that have been linked to, in some of the previous literature. For example, this alpha-1 acylglucoprotein has been linked to all these different diseases here, and hepatoglobin individually to these ones. So overall, glucan, detects the inflammatory component. But first time, now we have a clinically valid chronic inflammatory marker for veterinary medicine. So I think it's quite dramatic. Just very quickly, I will show how this performs. So we also had a cohort of samples for little obese dogs and then little lean dogs with maybe sarcopenia. And this is how it looks like. So we have this circle plot here. So the dogs, so there's a body condition score here correlated with the metabolomic profiles that we have. So different molecular groups are here in different corners of this circle. But in this outmost layer here, you will see the obese dogs, and you'll see that this metabolomic profile in this circle here is actually quite different from these dogs that are actually lean and have sarcopenia and problems with muscles. So again, the entire metabolomic profile is quite different. So if you have a dog come into the clinic, and of course, if the dog is very fat, you'll see that it has a problem. But sometimes, it's not very easy for owners to recognize, whether your dog is perhaps should go into some kind of diet or lifestyle changes, etc. What's important here is that you will see the glucose or the chronic inflammatory marker is really upregulated here. And we know that, for example, in obesity, there's a chronic inflammation behind the disease. So it comes up. So you should do something to really bump this down. We also measured very carefully the muscle condition scores, and here are the results. So if you really have bad problems, then again, you see that, the metabolomic profiles are quite different here. And there's a massive changes in different components. Amino acids are affected, of course, which is expected in muscle conditions here. So this is my summary slide. So I think, we have managed to validate very interesting new metabolomics platform for dogs with 123 biomarkers, and running through, various different cohorts gives you very exciting results, which tells you that you can perhaps start, developing these metabolomic fingerprints that would then enable, more personalized treatment optimization, etc. So I'm quite excited about this. Of course, this is very early of the story. So what needs to be done is really to collect large numbers of samples across different conditions and really try to understand what the metabolic profiles in these different conditions are. Before treatments, after treatments, really regular monitoring. So there's massive amount of work that has to be done. But again, like even in genomics, I think we have a fantastic new omics platform that could be used for the benefit of these animals. Thank you.